Hello, and thank you for joining us today for the webinar, Integrated CMC-based Pharmaceutical Development, Accelerating the Molecule to First in Human, which is brought to you by Invivo and sponsored by Syngene. My name is Andrew Warmington. I'm the Manufacturing Editor at Citeline, and I'll be your moderator today. Before we get started, there's just a few quick housekeeping notes. First, if you experience any technical difficulties, click the Test Your Connection button, and this will take you to a connection checker and FAQs page. You can also use the Chat Now function to reach our support team directly for help. We'll be holding a Q&A session at the end of the presentation, so please feel free to submit your questions at any time. Just type your question into the question window at the bottom of the screen and hit Submit. We'll answer as many as we can during the live session, and if we run out of time, we'll follow up with you via email. And yes, today's session is being recorded. A link to the on-demand version will be available within 48 hours. So in today's session, we'll be exploring how an integrated CMC approach can accelerate early phase pharmaceutical development, enabling faster regulatory filings and a more confident transition to first-in-human studies. Our speakers will discuss how aligning drug substance, formulation, analytical and regulatory strategies from the very start can mitigate risk, improve scalability, and ensure phase-appropriate quality across development stages. They'll also share how leveraging quality by design data provides deeper insight into material behavior and process control, supporting smarter decisions that carry through to later clinical phases. So let's get started. I'm pleased to introduce our speakers for today's session. We have Purushottam Singnakar, Vice President of Formulation Development and Manufacturing at Syngene, Hrishikevasa Reddy, who is a team lead and research scientist in pharmaceutical development, also at Syngene, and Deepak Mundra, who is an independent consultant. And with that, Purushottam, the floor is yours.
Yeah. Good afternoon, good morning, good evening, all. Now today we are presenting you a very interesting topic on how do we really accelerate the first in human development and get the molecule on clinic as quickest as possible. Before we going into the integrity of first in human development, I just want to just give you very high level update on who we are and what services we provide. As you know, Syngene is one of the global leading contract research, development, and manufacturing organization, providing services right from discovery services to development to a manufacturing and commercial launch of molecule. We are a fully integrated end-to-end contract research manufacturing organization, working on modalities in terms of small molecules, biologics, newer modalities like oligonucleotides, peptides, protacs, newer areas what we are ventured right now also into antibody drug conjugate and so on. So there's quite a diverse expertise as an organization driving solutions to the problem statements and bringing the molecule to the clinic as fast as possible. So as part of a drug product development, right, we have established quite diverse technologies here which can support taking molecule from late stage discovery to the first in human. As you understand, as we follow through the presentation and the scientific intricacies, the technologies that one need, depending on the molecule, is what are being established here at Syngene, starting with, say, enabling formulation technologies, specifically for molecules which are very low solubility, molecules which has poor viability, needs to really work on to improve the exposure potential for the molecule, and enabling technologies like spray drying, SDD technology, hot melt extrusion, nanotechnology, or liquid filled capsules, changing the viability of a molecule. Very extensively well established here at Syngene. Moving to the late stage technologies like modified release formulations, how can we transition formulation from once a day, multiple dosing to a once a day dosing or week dosing or a month dosing in terms of both orals and injectables. Also, very well established platform in terms of quick to clinic, which we are today's topic is to deep dive into a platform that we establish here as a quick to clinic. enabling approaches, how those risk-based decision approaches along with the phase-appropriate quality systems, understanding the regulatory and uh, pharmacokinetics and pharmacodynamic aspects of a molecule to really get a successful first in development. I will now hand over to Rushikeshwa to take it forward for the next topic for discussions.
Thank you, Purushottam. And so we are going to launch a poll question. Let us know your thoughts or early experiences in working with various phase appropriate pharmaceutical development. Thank you for sharing your point of view, audience, and it's been very responsive poll we received, and we'll go and start our introduction of our topic. So, as Purushottam mentioned, how the integrated CMC strategy will help us quickly going to the FIH and benefit the new molecules to be moving from discovery to development quickly, and enabling the small biotechs and large biotechs to get the benefit and reap the benefits of right or regional pharmaceutical development, and how we can design the pre-formulation studies, analytical method development, drug substance development, and various aspects of pharmaceutical development will be discussed today. And not only that, team will be discussing about various phase appropriate quality systems and regulatory guidelines briefly, and which are very critical for quickly moving the NC molecules from discovery to development to quick to FIH. So today's topics covered will be a brief workflow of the integrated CMC product, and followed by various key considerations that are considered for quick to FIH, and two case studies and summarizing what we discussed. So with this, I would like to hand over the next section to Dr. Deepak.
Thank you, Rishi. Hello, everybody. Happy to join this discussion. I work as an independent consultant in the CMC product development space. I've worked very closely with the Syngene team on a molecule to bring it to first time in humans. So I've actually seen this workflow come to life. So happy to join this discussion. So what you're seeing on the screen is, I'll call it as 4 swim lanes. So the top swim lane is what's called as a developability assessment. Typically, you might have a lead candidate or a lead candidate along with 1 to 2 backups. In the first swim lane, you're trying to assure that you have the right drug or the right candidate to progress into clinical development. So in the preclinical part of the development, you're looking at pharmacokinetics and ADME studies. And then in the drug substance, you're synthesizing your lead candidate using a medchem route. You also perform the salt and polymorph screening at this stage, followed by the pre-formulation excipient compatibility. And then you also have analytical method development in the same swim lane. So at the end of this at the end of the activity of this swim lane, you want to make sure that your lead candidate is suitable to progress into a drug product, which can then further be developed for clinical studies and then commercial launch. So that to me is a key step in the entire development paradigm for any drug to ensure that it is developable all the way to commercialization. The next step comes is the drug substance and the drug product development. So now you're actually developing a drug substance and drug product, which you can take it to the clinic and make sure that it's successful in the clinic. So you start with exploratory tox and DRF studies, which is to find out your dose range, which then leads to initiation of your 14 and 28-day tox studies, which are your IND enabling tox studies. Then you also have the drug substance development section in there. And then based on the drug substance development, you generate your drug substance lot, which then actually feeds into the initiation of the tox studies. At the same time, you're also doing drug substance and drug product lab stability. You want to make sure that you have a drug substance that A, it's stable for tox studies, and B, it's also progressible to as a clinical formulation. And then you also start your drug product development at the same stage. As much as possible in this stage, you want to make sure that your drug product is stable, both for tox purposes and also for clinical purposes. Then the next swim lane is where you actually make clinical supplies. So you have a drug substance GMP campaign, which actually manufactures your drug substance to take to the clinic. And then you're also doing drug substance and drug product method validation. And then the drug substance GMP campaign then leads into your drug product GMP clinical supply. So keep in mind that the sort of the third swim lane is what actually is now you're generating data to go into the IND. So that's the reason that the entire third swim lane has to be done in a GMP fashion, because this data is auditable by regulators. And then all of this then leads into authoring your regulatory dossier, which is, you have your GLP tox package, which goes into the respective safety module, and then all the drug substance and the drug product data goes into the module 3 of your IND or IMPD, or that's your opening IND or IMPD for first time in human. At this stage, like I mentioned, from the third swim lane, you've generated all the data as GMP. You also generate ICH stability. And then we'll also get into the details of some nuances and differences between US and UK, requirements for a first time in human. Or when you go to EU or UK, you also need a QP release, which we will touch upon in the next slide. So here are some of the key considerations for the CMC development, the drug substance development strategy, the regulatory consideration, the developability assessment, and so on. So we'll start with some of the top ones. Okay, so for drug substance development consideration, you can almost divide it into 4 buckets. The first one is synthetic route. So you want to make sure that you're not using any hazardous reagents. Your synthetic route is actually scalable, because keep in mind, this is where you're moving from a medchem route to now a chem dev route, which is then suitable to scale up. So that assessment is quite important here. You're also doing a genotox risk assessment to make sure that your drug is not genotox. You run some in silico models such as Derek and Lead scope, and also you run your intermediates through that genotox risk assessment to make sure there's no potential genotox impurities that can be found in the final drug product. Your route and solvent selection is quite important. Like I said, now you're moving from a medchem route to a chem dev route. So things like yield, purity of the material, they all start to become quite critical at this stage. And then you're also moving from a lab to a GMP area, where you're actually synthesizing the drug substance in a GMP fashion. Then when it comes to the target attributes, you have to assess salt versus free base. Again, Hushi will touch upon it a little bit more detail. What makes you choose one versus the other? There is also a solid state form, and there's a risk assessment to do that. Particle size distribution, and also the impurity profile for a tox and the GMP batch. These are the considerations at this stage. Also, in terms of like how much material you're synthesizing, so in terms of your lot sizes, there's some general references provided here, which is for a proof of concept batch, you're looking at 10 to 25 gram. You're also then the next step is your development or your reference standard batch, about 100 to 200 grams. At that point, then you're starting to scale up from what's called as a gram scale to now kilo scale. So you go into a tox batch, which is 1 to 2 kilo, which supports your a 14-day and a 28-day tox studies, which supports your opening IND or IND enabling studies. And then followed by your GMP clinical batch, which could be in the 2 to 4 kilo range. Some of the quality and regulatory considerations are regulatory starting materials in terms of what you eventually intend to file as a regulatory starting material. It's a consideration in the early stage of development. Then the next one is single lot versus split lot. This is a bit of a controversial topic, I will say, within the industry. Some people like to make a single lot, which can be used for both tox and for GMP purposes, so that the impurity profile looks very similar and you don't have any new impurities in the GMP lot. Versus some people like to actually make a split lot, meaning you make a large lot and then you actually have a dirty lot, which is what's called as a dirty lot because you want to make sure you qualify your impurities in your tox study, and then you purify a sublot to ensure that you have a cleaner lot that goes into the GMP step. Again, there is no right or wrong answer. It's just a style and preference of different companies and how they operate. As long as you make sure that the impurities that go into the clinical studies are qualified, and you do not have any unqualified impurities that you will struggle with the agency to justify. Then comes your batch records. You have to make sure that your batch records are quite flexible. It's because it's an early stage development. Uh, you're still making your first lots, so you want to keep it more flexible. And the specifications have to be phase appropriate. My go-to guidance is the ICHQ 6A. I usually refer back to that and assess whether some of the requirements are a pharmacopoeial requirement versus a regulatory requirement. And also, as much as possible, you want to follow the pharmacopoeia other than your drug substance related impurities, where you would need to have a specification which is based on your qualified impurities. All the other testings and specifications, it's preferred to have a pharmacopoeial method rather than developing a product specific method at this stage of development. The next. Okay, so now in terms of regulatory considerations, so of course you can go to the US for an opening IND, or you could go to EU for an IMPD. You should make sure that you read the guidance documents specifically about the toxicology guidance document and qualified impurities. How do you translate the impurities from your tox studies into your first time in human? There's a lot of considerations also in terms of your starting dose in the clinical study as well as the top dose. That's usually what's negotiated with the FDA at this stage of development. Your batch release is performed by QA in the US, whereas in the EU, it's actually performed by a QP or a qualified person. You can have a pre-IND meeting depending on your indication and if you have any specific questions for the FDA, whereas on the IMPD, you have to go through a scientific process. The process is very similar. It's just a different name, and you have to be cognizant of how long it takes, whether it's a type B or type C meeting, and then how long it takes for you to engage with the FDA on this. And then the labeling requirements are also different. The labeling requirement, I'll specifically point out, like one of the key differences in the US, you don't specify the shelf life. Your shelf life is managed internally, your shelf life or expiry date. Whereas in the EU, you have to file for the expiry date, which is actually approved by the reviewer. And I can tell you, you get a lot of questions from the EU based on my past experience with regards to establishing a shelf life. At this point, I will hand it over back to Rishi. Thank you.
So audience, so Dr. Deepak has given some background about the regulatory aspects and how a workflow should be there for an integrated CMC. So once we are having some workflow established, so in the formulation point of view, and pharmaceutical development point of view, there are a lot of developability considerations to be considered in the stage of even the potential candidate selection, or in the late discovery stage where there are shortlisted candidates, which would be the primary of importance. Where these are few considerations like physicochemical properties, like what would be the ionization constant, what is the intrinsic solubility, which is very important to molecule to have their sync conditions established when we are taking orally, especially. And what would be the right salt versus what would be the right form that would be required to present further. And going further for the biopharmaceutical properties, definitely not only the solubility, the BCS classification or DCS classification, which I'm going to briefly speak about in the later slides, where the permeability and the ADME point of view is also one of the important factors which you have to consider. And the next point, which would be important on the formulation point of view and the oral bioavailability point of view, is the positive or negative food effect of the compound, which is also impacted because of the molecular properties too. And last thing is the oral dose proportionality. Even though we are getting the required percentage F or the absolute bioavailability in the lower doses, we have to see if you are getting the desired oral dose proportionality when we are increasing the doses to achieve the MTD or the tox margin, and select the human equivalent dose further. So before going to the formulation and various aspects of it, so the starting point here is the solid state characterization or the investigative characterization part of the drug properties on the solid state perspective is also important, which Syngene does. And with few milligrams of compound itself, we can establish what would be the crystallinity, what would be the polymorph of the current discovery scale molecule. And hygroscopicity is also very important at the earliest stage to identify and quickly screen which type of form or salt should be selected, where let us say there is a free base and it is showing poor solubility. In that case, definitely a salt can show a solubility enhancement benefit, and also in the per se of the PK, it will be further enhancing the bioavailability because of the solubility enhancement. So it's not only the criteria, but bioavailability is also one of the point of view. And let us say if it is a BCS class 2 molecule, even the particle size plays very significant role. So even though at the earlier stage, better to understand based on the DCS classification, which I'm going to explain briefly, and understand the particle size impact at this earlier stage only. And amorphous solid dispersion tendency also is one thing which we have to establish at very quick stages. And that's where we understand the glass transition temperature of the molecule, etc., and how it may impact on the kinetic solubility of the molecule. So this is what I was speaking about, the DCS assessment or developability classification system, which we know as BCS classification system as per the US guidelines, where 4 quadrants or 4 types of classifications are there, where it is more about dose dependent and very specific about the highest volume, that is 250 ml as per BCS. But the DCS is where we don't know for the new molecules what would be the highest dose, and can give what would be the desired or the desired or right path forward for the formulation selection strategy. So if you see here, there are various pointers coming over, where in the quadrant 1, where the viability is good, where the permeability of the which we establish using the CAGO2 permeability studies will be done. And when the dose versus solubility ratio, where we see for single ascending dose or multiple ascending dose, or even for the preclinical studies, where the doses might be going from the lower to higher. Based on these assessments, we'll be dividing these into 4 quadrants. And if it is highly soluble and highly permeable, where the solubility is meeting as per the dose versus solubility ratio criteria, it goes into the DCS1. And if it is 2A, which is in the navy blue pointer, it is considered as a dissolution rate limited, because it has some intrinsic solubility, but probably when we are increasing the doses, probably it is not giving the desired availability or dose proportionality because of the dose limited or dissolution rate limited absorption. So going to the orange pointer, where it is in the quadrant of 2B, the solubility limited absorption is considered, where the permeability is good, but it is the classification will be wholly because of the poor solubility of the compound. And we select few types of formulation strategies, which I will be explaining about the right formulation strategies for each of these quadrants in the next slide. So in similar way, class 3 and class 4 will be subdivided based on the poor permeability and good solubility for class 3, and DCS4 for poor solubility and permeability. So this is what I was speaking about. So once we assess the dose solubility ratio criteria and understand what might be the lowest and the highest doses, so there are two criteria. One is which formulation might be right for preclinical, and which formulation can be quickly translated for the FIH studies. So this is where we have given a few examples here. So if it is coming under class 1, for giving as an example, we can go as simple suspension or a solution by just adding water, since it is showing desired solubility. And that can be quickly converted into any type of formulation which we mentioned here, like any drug in capsule or any drug in bottle, and constituted with a ready-to-use vehicle which are marketedly available, like Ora Suite, etc. Or it depends on organization policies or requirements, or thought processes too, where we can convert into unit doses at the early stages also if required. And the major brick-like or grease-like molecules, they fall under 2 or 3, where we convert them into nano suspensions or solutions in preclinical stage, and quickly convert by using various approaches which I mentioned here, like micronized API, or using some solubility enhancement agents or dissolution enhancement agents, and converting into unit dosage forms. That can be done quickly. And lipid-based drug delivery can be used if it is even in the class 2B also, and also going to the lower scales of classes, where class 3 and class 4, where self-emulsifying formulations or lipid solutions can be given, and for preclinical readily, and that can be quickly converted into liquid fill capsules when we are going through the phases of development. So this is a brief explanation about the strategies which we can think about from preclinical and translating quickly into the clinical formulations. And there are some points where we mentioned as DIB here, where we can use the facility of phase 1 clinical facility, and go with on-site compounding facilities too. So these are few considerations, not only about solubility, when we are developing any formulation or preclinical formulation, to be very specific. We have to consider various aspects like intrinsic solubility, which I mentioned about. Is it a flex substrate? And what would be the pH effect? And will it be crashing out? Even though, like, we have seen in multiple cases, like if it is a solution, then we might be expecting the oral bioavailability will be as per the desired exposures, but we are not seeing the same exposures. The probable reason might be because of few reasons, like flex substrate nature of the compound, which we understand at the early stages by performing the CACO2, and also PGP flex substrate testing. And also one more thing is the food effect, as I mentioned. So a simple study of fasted versus fed can give us more information on that. And also to understand the difference between the suspension versus solution, that is also one kind of PK recommendation which we commonly go with, where we'll understand if there is a variability in the suspension versus solution PK study. That is also one thing which we consider. And the purpose of the study is one thing, where we might be using some excipients for a single PK or dose proportional bioavailability exposure purpose, but we have to think about what would be the safety of those excipients when we are using for repeated dosing for GLP tox studies. And animal species also one of the thing, which I think some of you might be knowing, that if 280 is being used, it causes nausea or vomiting in species like non-rodents of like dogs especially, and we have to take considerations similar to that. And that's it. I think these are few considerations. And apart from that, when even though we are developing a solution, we have to see what would be the fate of it by performing some in vitro dilution studies too, where we understand if the solution can withstand without crashing or not. And these are few things which we have to consider before developing any formulation. So that's my point on the preclinical considerations. So let's launch a second poll question before going and handing over to Dr. Purushottam to speak about drug product considerations. thank you for your valuable inputs and point of views. It's been a nice response from audience, and I would like to hand over to Dr. Purushottam. Dr. Purushottam, thank you.
Thank you, Rishi. Thank you, Deepak, for taking us through the integrated development and what different aspects one needs to consider, right from developability assessment, drug substance, preclinical development. Now, I'll just make you make the details through the formulation considerations, what one should look into working on first in human strategies. So what are the first in human formulation considerations when it comes to formulation aspects of it, right? We understand early preclinical formulation aspects, right, from physicochemical properties to some few PKPD understanding that could coming from a early assessment of the molecule, and how do we really take that into effect in defining what should be our formulation strategy. So putting all data together, what we generate as a part of our developability assessment, looking at the molecule lead compound where we understand solid state properties and biopharmaceutical properties as part of our development, whether this molecule makes sense for development in what form, right? So all those physicochemical data, permeability data, especially COCO2 permeabilities at early stage, solution versus suspension PK studies as part of a early development or preclinical development, sometimes we end up in understanding is really a low solubility means going to be a lower exposure of a compound. Many a times it may not be the case. So how do we really understand that in defining a right formulation development strategy, which is first, which we can get to a successful first in human clinical supplies made on time. So, looking at the drug product development considerations, we look into four buckets, right? One, physicochemical properties, dosage form selection, clinical requirement, and quality and regulatory. If you look at these four buckets, these are not independent four buckets when you define a strategy for formulation development. All this has to be look in consolidation with each other to really understand what are the interacting aspects one should into account, right? So very first one is physicochemical properties like solubilities. We discuss on uh, uh, solid state properties, hygroscopicity, and all physicochemical parameters. How do one has to really take that account into is the API drug substance is going to be hygroscopic? How is the solubility studies defining at different pH? Is it pH dependent solubility? How is if it's a low solubility? How is being solubility enhancing effect has happening in terms of fast fed state SIF and fasting state SIF and any solubilizers, right? So once we understand solubility aspect for the molecule and the physical properties linking it to the formulation requirements, we also have to understand from a dose form selection perspective. How do we look at this form? What dosage form you would really make sense for the molecule what we are developing? One has to really look at it into developability classification, what Rishi has taken us through, depending on the solubility and permeability. Also, one looking into the MTD dose, what we are expecting and what the dose strength we are anticipating for clinical first in human phase 1 clinical study. Based on that dose range anticipation, we define what should be our formulation, right? Whether it can be a simple drug in capsule, as if we are really have physicochemical properties of a drug substance supports filling up a capsule into a drug, achieving the quality parameters, one can quickly select on a drug in capsule. Drug in bottle, on-site compounding, capsule, formulated tablets, capsules, as well as enabling formulations when there are solubility challenges, one has to look into. Uh, so coming back to really selection of this dosage form, also many people depends on their past experience. They take it a comfort level, right? Many organizations have their own strategy, a fixed strategy of a formulation to take it for FIH phase 1 development. There are organizations who believe more doing into formulated tablets or capsules. There are organizations which believe in drug in capsule to just quickly move on, or formulated powder into a capsule. So it depends on how each organization and strategy they have been going through. But especially for enabling formulation technologies, one has to be very, very careful in selecting the formulation strategy for enabling. Very first question one has to ask is, do we really need enabling approach for the molecule? And that can be very well understood through solution versus suspension PK. That can also be understood through some of the early preclinical tox studies has and the imperative pharmacokinetic and pharmacodynamic data what showing us. All those has to be taken into consideration. And there's still, if any ambiguity remains, one has to quickly look at how can I conduct PK studies on non-rodent animals, right? It can be dog PK or mini pig studies to really say that yes, the formulation strategy I'm selecting is going to be right for first in human development. The ultimate goal is how I can maximize the exposure of a compound to understand safety aspects in a phase 1 studies, and that's the ultimate goal one has to keep in mind when selecting formulation for first in human development. And then again, from a regulatory perspective, yes, how do we really define the phase appropriate specifications, uh, complying to ICH guidelines and US FDA regulatory guidelines and EMA guidelines from that perspective, right? And of course, as we enter into formulation development and then moving to of clinical batch manufacturing, of course, then one has to follow all GMP requirements that are required uh, as per the regulatory norms. So in short, one has to be, say, spending more time in terms of understanding the molecule, understanding what different types of data are available, and what kind of a risk one should take to define right formulation strategy for that FIH. So coming back, say we were looking into say different types of formulation strategies, what Rushi has presented in previous slide, right? Drug in bottle, drug in capsule, tablets, formulated tablets or capsules, enabling formulations, or lipid-based capsules, right? Each one has their merits. Each one you cannot just blindly apply these formulation strategies for any molecule. And as I said earlier, one has to understand both biopharmaceutical risk assessment as well as form risk assessment and the process and the dose range response understanding for the molecule, right? And based on that, one can select the strategy for first in human development. Uh, Drug in capsule is the most widely used option for formulations. It really a molecule support for directly filling into capsule, and we are able to get the highest exposure of a compound based on the PK studies what we understood. Uh, but here, drug in capsule, now one has to be also very careful. I just share you one example here. It appears to be so simple that filling the drug and getting the capsule and get the first in human quickly delivered. No. Especially molecules which are hydrochloride salts, molecules which are two hydrochloride salts or tartarate salts poses many times a problem when you are defining drug in capsule formulation. Especially when they absorb moisture, they tend to form a gelling layer around the drug particles, and then you get the slowing down of a dissolution and so slowing down of a exposure of a compound. So one has to understand all these aspects. Just to give you example on that, many a times hydrochloride salts try to dissolve the gelatin capsules, and sometimes you find that my capsules are just completely crunch, right? So think on all aspects, really and deep dive into it before selecting the strategy for formulation development. And of course, the formulated tablet capsules, which as you go are very common in terms of enabling the flow properties, enabling the surface wetting of a drug substance, enabling solubility, dissolution profile enhancement. Those are very, very common and age-old understanding as a part of formulation. But how we rightly apply each at a different stage is very important. And one aspect what we have learned through a number of programs that we successfully delivered and the learnings that we have adopted is is when you arrive at a development strategy, it's not a one-person job to arrive at development strategy. The best way to have a right successful approach in shortest time span would be to forming a subject matter experts group for a given integrated program. So there are different experts coming from drug substance, experts from drug impurities group, experts from formulation group, experts from analyticals, experts from uh, PKPD and so on and so forth, and all the team dwelling together the data what has and what that strategy actually means for enhancing the exposure of a molecule. Now, we were looking into formulation strategies, but equally important is how we look at the analytical development considerations when we are developing the molecule from late stage discovery, preclinical development, first in human development, and GMP manufacturing and clinical supplies for phase 1, right? Every stage of a development analytical becomes a part of it, and very important, should not be ignored. And how phase appropriately you are looking at analytical is also become important. So from our experience, putting the analytical scientists and expertise from drug substance and drug product together at early on at a very preclinical development stage helps you to address many challenges what you could see afterward coming from, right? So API expert, API analytical expert putting thought process from a API process perspective, and the drug product analytical expert putting perspective from a formulations what and what can interference come on. So always should be a approach that how can I come up with a common analytical method for both drug substance and drug product. Very, very important in terms of saving the time for first in human, because as you are moving from a lab scale or a route of synthesis in chemical development to pilot scale and moving into GMP manufacturing, there are lot many process changes to adopt. Complex chemistry gives you lot of many challenges in terms of some new impurities getting detected or something unknown impurities finding the way which you really do not want to spend too much time on to get the first in human as quick, right? So how those analytical methods cannot discovering later on at a drug product stage, and how that common method really able to put finger point on, yes, this RRT means for this and this RRT is going to mean for this, rather than I finding and synthesizing each and every impurities and trying to characterize and spending time, sometimes significant time in the CMC development. So how you approach it is also very important, and that's how we have learned in analytical space that how do you really bring the expertise from drug substance, drug product perspective together at right from early on to the phase 1 delivery. So as we were dwelling on scientific aspects of it, right? What do you look into developability assessment? What do you look at it when you are at a preclinical development? What do you look at it when you are a first in human formulation development? And in parallel, what do you look at it when you are scaling the drug substance from a lab scale to GMP scale, tox lot to a first in human lot manufacturing, right? So in all those aspects, the governance becomes very, very important when you drive such an integrated program to reach to a successful outcome. Bringing in, as I said earlier, bringing in the subject matter experts of the interdisciplinary team together, forming a core team group led by a CMC lead who has kind of the technical lead for the entire project, and then connecting each subject matter expert at different stages and which driving the decisions at each stage gate of a development, right? That becomes important, and that's how we actually form a core CMC group led by a CMC lead in driving the technical complexities as we run through, as you learn and as you go through the development. In parallel, it's also very important how are you managing non-technical activities and falling in place at the right time and at the right stage, right? So there will be an integrated project manager defined who takes care of from a typical project management perspective and work with the CMC team and the cross-functional subject matter expert group core team in driving the project. And it's very, very important from a governance perspective, how do we really bind the teams together uh, to save the time on to for a faster quick to clinic. So here is the same what I said is in a pictorial presentation that how the CMC lead forms the core group and the experts connecting to it and the project manager driving the project management activities, communications, logistics, arrangements, all different aspects of it. So basically, in this core team, one also the core team members from a regulatory team, which I did not speak on. So regulatory expert would also be joining as a part of a core team, and very important to really engage regulatory expert right at very early preclinical development stage, right? Because many other aspects starts getting integrating from a regulatory standpoint, uh, and defining the strategy from a regulatory compliance and regulatory requirements perspective. So thank you for silent listening. Now I would hand over to Rushi to take us through the case study on one of the case study on the integrated development where we moved molecule from late stage discovery to first in human. Over to Rushi.
Thank you, Purushottam. So, audience, we have been hearing from all the team, our panelists, from like Dr. Deepak, Purushottam, and myself, that various types of considerations we can consider usually in various stages of development. So, this one case study, which we had successfully delivered with integrated CMC approach, where we started the developability assessment from the lead selection stage itself and selected a right candidate, and then performed salt screening, polymer screening, and in parallel, we performed some pre-formulation studies as workflow shown by Dr. Deepak. And while it is happening, we select the suitable salt and the right form of the salt. And once we selected that, we have gone to the next stage of preclinical formulation development aspect, where we quickly connected a suspension versus solution PK study and understood this definitely a solubilization approach is required. And then we have performed dose proportional PK studies and observed the dose linearity, and then selected the suitable or the lead formulation which we prepared as a solution. I remember it was using a complexation approach, and based on the MTD studies and followed by the actual GLP studies, we could derive the human equivalent dose to start for the phase one, and to facilitate quick transition into the FIH while the scalability and the process development is happening in the drug substance point of view. We developed a drug in bottle approach, which I was mentioning about, where it was a two bottle approach where one bottle had API as required to fill to serve the various doses or the wide range of doses that is required for the SAD studies or single ascending dose studies that are starting point for the phase one. And the second bottle had cyclodextrin, where at side the pharmacist will be compounding with water and mixing both the contents of the bottle and to prepare a solution. And definitely palatability was also evaluated in parallel with e-tongue assessment, and where we found because of the complexation the palatability was better. And there are other approaches which Dr. Deepak will be mentioning about alternatives in the next case study. And once we have transitioned into the phase one, while the studies are happening, we discussed about how to transition this quick to FIH formulation, which we developed, into a more commercially viable option, or the uh, formulation presentation. So that's where we have discussed and tried to convert into a pellet kind of formulation. Why? Because still at the end of phase one, we need to conduct various types of repeated dosing in the multiple ascending dose studies, and we don't have or we may not be having proper idea to what extent of lower dose or what extent of higher dose or the maximum dose that is required for the safety and the efficacy of it when we are going with the repeated dosing. So to make it as a flexible dosage form, we designed a IR pellets and did a bridging PK study in mini pig, which was more suitable non-rodent species instead of dog, where dog had some limitations, which you might be knowing. And based on the mini pig PK study, we have confidently gone to the clinical dosing, and we found the required oral bioavailability or AUC exposures. And that's how we have been successfully delivering other programs too. And this is one classic example. And uh, and I would like to hand over Dr. Deepak for presenting the next case study. Thank you.
Thank you, Rishi. If you go to the next slide. So I think as you've heard from Purushottam and Rishi also, that there is key considerations with regards to your phase one formulation. It's balancing the speed versus bioavailability, right, and the quality of the product, right? So typically your phase one, SAD, which is single ascending dose, and MAD, multiple ascending dose study, it require multiple doses with the goal to maximize the exposure. Now, in terms of approaches, as we heard some of the examples, right, like a direct drug in capsule, and you saw examples from Purushottam, and based on my experience also, there could be some issues which could be drug specific related, whether it's a particular type of salt or it's the wetting property of the drug. That could lead into bioavailability issues for a drug in capsule. So again, everybody has their favorites, but I think this has to be driven by the molecule properties. So this comes back to science, right? So uh, my favorite approach is actually having a fully vetted or a solubilized drug product. So whether it's a suspension or a solution, and you can make up to like three strengths: a low, mid, and a high strength. Uh, and the couple of reasons I'll explain. A is if it's a suspension that's fully wetted, then you don't have any concerns about wetting issues or not having any excipients in the first time in human formulation. If the drug product is fully solubilized and if it doesn't crash out, as Rishi explained, then you're assured that the dissolution part has been taken care of. That means the only aspect when it's ingested is about the permeability and then getting absorbed. And then I say three strengths specifically because when you go into a SAD and MAD dosing, your dose range is pretty high. So your starting dose could be as low as half a milligram, and your top dose could be as high as 500 milligrams. So it's not one size fit all. So that's why I always recommend doing a low, mid, and a high strength so you can bracket the range of your dosing scheme in your first time human. Some of the other challenges that you can face, and this is actually based on a real case study, which I have actually dealt with, and I've actually worked with Syngene to answer some of these questions actually that we even got from regulators. So one of the challenges that we faced was the microbial quality, because in first time in human, you're not actually adding a preservative, because it's a quick to the clinic kind of approach. So you might get limited for your in-use period to under 24 hours because the microbiological quality of the drug product has not been fully established. And also, it is recommended to store the product refrigerated from manufacturing to dosing, and it should be dosed within 24 hours. You have to establish a hold time or an in-use period where you establish the assay and the impurity profile at refrigerated condition and also at room temperature condition. You have to make sure that your dosing is accurate, especially when you're dosing a suspension. You have to make sure that let's say if it's a 1 milligram dose in a bottle, you have to make sure that that entire dose is delivered. One of the easy way is you make it a drinkable suspension and then you rinse the container, 1 to 2 times to make sure that the entire dose is delivered. And you actually do that as a simulation in the lab. So again, this is one example where I've worked with Syngene to demonstrate the dosing accuracy as part of the in-use testing. And again, it's part of it is using calibrated syringes and dispensers. So you try to make sure that whatever you're using in the clinic is actually what is used during establishing your in-use studies, which goes into the submission. The one more aspect which I think is ignored at times is the taste of the mouthfeel, because if you're actually dosing a suspension or a solution, again, the product that I worked on using the e-tongue approach was extremely bitter. It was actually as bitter as close to quinine. So it was a major concern during the dosing that people might have nausea or vomiting if you're asking them to drink this suspension or solution. So one easy way was actually using a Listerine strip post dosing, and actually what that does is it actually numbs your tongue, and you don't get the aftertaste. And it can also be used as a blinding tool if you're in a blinded study. Also, there's also actually an aspect of a bitter tasting drug which carry bitter tasting excipient also, which you can use if you're trying to blind your dosing. And then another consideration is the salt disproportionation. So, making sure that your salt doesn't disproportionate, or there's no solid state changes from the drug product to the in vivo, right? So, for example, making sure that you test your drug in the presence of simulated gastric fluid, as well as FASF and FSF, to make sure that if you're selecting a certain salt, it doesn't disproportionate when it comes in contact with the gastric fluid, and then all of a sudden you lose bioavailability. So those are all the considerations you need to keep in mind. Very much on the right side of what you're seeing is actually an almost a generic way of presenting the same case study, where you have the pre-formulation profiling of the compound. You perform your solubilization trials where you select your formulation, your excipients, your formulation vehicle, and then you identify the vehicle for your drug in bottle presentation. And then the drug in bottle prototype is developed at that point in terms of the volume of the vehicle, the reconstitution time, and like I mentioned, if you have 3 different strengths, you have to make sure that you simulate each of those strengths in the lab, and that data then goes into the regulatory submission. And then the recovery and the rinsing studies that I've given example of how to make sure that you're delivering the accurate dose. At the end of it, all of this actually becomes a master batch record for the on-site compounding at the clinical study. Again, this is an actual case study of what I have worked with Syngene, where they actually drafted a master batch record for us, and I was able to reproduce that to the clinical study site. I was on site and made sure that the manufacturing process is followed, and then we did a testing to ensure that the drug product meets all its critical quality attributes. So hopefully with this example, I was able to show you how you can get to the clinic fast and quick, and also you're not losing or you don't have any oversight of bioavailability considerations. So some of the advantages in partnering with a CRO like Syngene or any other CRO, It's a one-stop solution, actually. So Syngene, for example, can support all your programs from early discovery all the way to commercial supplies. There's all the other considerations as well, but I will actually focus on the one-stop solution. Like I said, I've used them in the past for a program which came out of discovery, and then we've used Syngene to do all of the drug substance solid state screening work for solid state form screening, followed by all of the tox formulation and the clinical formulation development, including all of the analytical work packages and everything that needed to go into the submission. So that's what I mean by one-stop solution is they can help you guide not just that part, but also the preclinical studies in terms of the tox package, as well as helping you with the regulatory authoring and guidance on that as well. The other thing that I will probably highlight is the technical expertise. Again, um, formulators or CMC, personnel have their own niche area, right? So you can be a drug substance lead, you can be a drug product lead, you can be an analytical lead, but you cannot have one person that will know every aspect of development. So this is where I think Syngene has a strength that their team works as an integrated team, and you can really rely on them to provide their expertise in all the various nodes of CMC development, like drug substance, synthetic route development, your formulation development, your analytical method development and validation. So it's really a one-shop stop that you can rely on. At this point, I will hand it over back to Purushottam.
Yeah, thank you, Deepak. Really interesting case study and the learnings really truly depict how one should be looking at as a quick to clinic approach and mitigate all the risks. Really, a really good case study on that. I would like to just summarize on the learnings and of course these learnings are just not coming from one or two case studies, right? These are the learnings which are coming through multiple integrated programs we delivering at Syngene, right from discovery development to the clinic, right? So very first one, and of course, the implementing these learnings into upcoming programs actually gets you the way forward. If I start with the developability assessment, right, one should really focus more on understanding drug substance physical and biopharmaceutical properties, not to rush, not to just make hurried decisions or uninformed decisions. Any decisions what you make should be very informed decision, maybe risk-based decision, but it should be very well understood, because that's where the things changes when you see at a later stages things are not falling in place, right? uh, the other one is in terms of salt form, physical form, and other aspects of it, right? When one is looking at developability stage and solid form understanding or salt understanding or physical form understanding, the target is to not to get a polymorphic form which gives me always highest solubility. We will not select metastable form at the early stage development and end up problems in later on. So always approach would be to look at what's the most stable polymorphic form and what those implications one should understand from a form assessment perspective. And same the salt, I think very good case study Deepak explained in terms of salt disproportionation, salting out effects, very important when you select the salt. Many of the point you might see that improvement in the solubility just because of the salt and as compared to the base, but some many cases you will end up into base having higher exposure as compared to the salt form. So just make a complete deep dive into understanding what are the biopharmaceutical risk and what are the uh, solid state or form risk of a molecule. The next important point, learnings from multiple programs, and that would help uh, as an audience for all of understanding is from a DS development perspective. Development is always focused on process, physical form, particle size, impurities, quantities, right? Appears to be very normal that, okay, one has to follow through all these things when you are developing a drug substance. No. Especially when you are doing initial solid state characterization of a molecule at developability stage, or when you are doing this solid state characterization at early discovery compound, which could be say 1 or 2 gram of a compound, or even sometimes 10 milligram of a compound if you are lucky. Understand those maximum information from that to save on the time and define what you would like to take forward. The second one, monitoring the process changes, impurities as you go on from lab scale to manufacturing. There are physical forms changing, there are morphological changes happening, there could be some new impurity coming in. How do we understand those risks? How the formulator also integrated with the drug substance chemist together when some process scale changes happening, and what that collective decision to take forward, and not just getting stuck in terms of, oh, what do I do now? This API is not flowing, or this API just got stuck, or this API got into polymorphic impurity, contamination, not a contamination, but I got some new polymorph, 10, 15, 20% of a new polymorph, and I have some other implications coming out of that, right? So as I said, integrated team is very, very important to really have a quick and successful outcome from integrated CMC development. And same thing, analytical, I dwell on how one should look at it, integrating analytical right from the preclinical development to the GMP clinical supplies. The other thing in terms of, say, like drug product development, one should look at it from a PK studies perspective, right? And especially very important when if anybody looking into enabling approach or anybody working on enabling approach, what kind of animal PK studies really makes sense to get a right outcome. I tell you one example, and I think that case study is not being presented here, but that was the enabling approach where team work on enhancing the solubility of a drug through solubilization approaches. All different co-solvent surfactant combinations were being formed, and then and the viability improvement was seen on the rat model. And then everybody was, oh, yes, this approach would work. Doesn't work that way. When we further tested this into higher animal, especially mini pig, the things actually we seen completely reverse trend. So very important to understand how the pharmacokinetic profile is likely to vary, what how different animal models can reflect into different outcomes needs to understand, and what should be the way. Sometimes not spending time on rodent animals to do some PK studies and things that are part of early stage development, and quickly jumping to do a non-gene higher animals like mini pig, dogs, and depending on the molecule and depending on the formulation strategy, that becomes to a very quick approach and robust approach to get a right selection of a formulation. And of course, the quality and regulatory aspects to engage early on in the program. So for example, if you are looking IMPD filing, how do you really engage QP early on so that you don't spend time later on to have all the formalities completed, QP audits, facility qualifications, product release for systems, processes are already aligned before you really start manufacturing the batch, and then just getting the quick QP release. So timing of each activity, integrating those timings becomes very, very important when one look at it, how to get a quick to clinic. Yeah, so thank you all for us listening to all the technical details what we gone through, and I'll hand over back to Rushi.
Yeah, thank you, Purushottam, for valuable insights, and Dr. Deepak. Anju, we are open for audience.
Great, thank you all very much. We have several questions from the audience, so let's jump straight into them. First one is, how will salt versus free base form in fact impact a toxicity study? Deepak, I think that'll be one for you.
Sure, thank you. So yeah, the salt selection is actually extremely important in early development, as early as your first DRF study, because what you don't want to do is change your salt in the middle of the DRF study or going from the DRF study to your tox study. The main purpose, if you think about a tox study, is to establish any toxicity related to exposure. And obviously the salt will impact exposure. So for that reason, your salt selection should be as early as your DRF study and taking it all the way to the clinical study, because you don't want to lose the link between your salt and the exposure, which is the basis for your dose selection for your first time in human.
Thank you. And second question, I think this will be for Hershey. How much stability should be considered for tox formulations?
It's a nice question because a lot of teams have a lot of different types of opinions on that. So there are three major criteria for this. One is to consider the actual stability of the formulation which we prepared for the PK studies, and how stable they are. Will they be stable for long term, or do we need to freshly prepare? And second criteria will be the capability of the tox team, the where the tox study is actually happening, where if the tox team has feasibility or the facilities to manufacture or prepare those suspensions or the preclinical formulation that will be required for the tox studies. And the third criteria is, even though they have the feasibility, it should be easy to prepare, or it depends on the complexity of the formulation compounding or the instructions which we are manufacturing with. So these are few criteria which we have to consider before taking any formulation for the tox studies, and then based on the stability, we have to decide how to proceed further. Thank you.
Thank you very much. And on that note, I'm afraid we've run out of time. I'd like to thank our speakers Purushottam, Hershey and Deepak for that great conversation, and our sponsor Syngene for making this event possible. On behalf of Syngene and Sightline, have a productive remainder of the day, and thank you for watching.